Hope you're doing well. Sorry, it's been a little bit of a while since I've posted my last lecture. In the meantime, we've had our midterm, and I've graded all those and emailed you back if you had attached a file, or just put the grades in the comments if you shared a Google Doc with me. So it should have sent you an alert, but if not, just go ahead and take a look. Let me know if you have any questions. I thought in general the exams were very good. The average was quite a bit higher. I think over 10 points higher than the first one. So nice job. It's been a challenging time, and I hope you all are doing well. We're getting towards the end of the semester, and we'll be done before you know it. So good luck getting finished up. And if you have any questions on our class, again, I encourage you to visit in our Google Hangout time, or send me an email or something else if you want to connect that way. So we're going to be talking today about environmental health and toxicology, and I have a picture of a bluegill sunfish here, which will be important in just a little bit. But I also wanted to just check in about Moodle. I sent out an email, but we have a new assignment on Moodle since we can't do our celebrations of knowledge anymore. I know that's a real bummer for you all, but we have these new assignments. This is a screenshot from our class Moodle page. Those green puzzle piece icons are these what they call hypothesis activities. So you click on that activity, it'll take you to a PDF of an article. And then I've asked you to provide at least 2 annotations where you highlight a little piece of that article and then write a note in there. There are instructions on there that'll show you how to do it. But just FYI, our first one is due on the 16th of this month, just a couple days. And then there's 2 more. So be sure you have that on your radar to go onto Moodle and check those out. So there they are, those 3 assignments. So every now and then I find funny articles that I like to share, and here's one on eagles that were running up their cell phone bills. So these authors were trying to study migration of these eagles, and so they put this SMS texting device. They fitted it onto the eagles' backs, and so this device would send a text message that contained GPS coordinates of where these birds had been, and it would store the data, and then when they got near a cell phone tower, they'd send all these messages that would download to a server. Well, the funny thing is, these birds, one of them, this eagle, went a long ways away from where they'd expected it to, all the way into Iran. And when it came into Iran, it was next to some cell phone towers that were owned by a service that had really high roaming charges. And so these researchers got this incredibly high bill for thousands and thousands of dollars for this cell phone bill, and blew up their project budget. But the good news is these cell phone companies actually gave them refunds and then offered discounts for their study moving forward. So anyway, I thought this was interesting when I heard about it. All right, so we've talked a little bit about the environment as a beautiful place, right? And if we have an aesthetic or spiritual connection to nature, probably a lot of us feel that at some time. And maybe there's no greater evidence of that than to show the positive health effects of being in nature. This was a study done, published in 2018, where they asked these participants in Europe a questionnaire about their mental health and also about their commuting habits back and forth in their job. What they found was really interesting. They found that those folks that commuted through natural environments daily had on average a significantly higher mental health score compared to those who commuted through natural environments less frequently. And the mental health benefits of traveling through nature were even higher for folks who reported what they call active commuting, so walking or cycling. So hopefully this is an incentive, if you're able, in a healthy way, to get out, get some fresh air, go for a walk, enjoy nature a little bit. But the environment can be a dangerous place too, and I mentioned in the beginning of class that issues like pollution are really what started the field of environmental science. Got people about how the environment serves is our habitat, right? How it provides us with resources, accepts our waste, and what it looks like when it's not able to do that naturally anymore. So this leads to the field of environmental toxicology. So this is the field that studies the negative impacts of some of these agents on living organisms, and most often humans as living organisms, but studying the chemical, physical, and biological agents that can have these harmful effects. So an example of chemical would be pollution, chemical pollution that gets into the ground and can get into the drinking water, or be taken up by the plants that we eat. Right? So those are examples of chemical agents. Biological agents, we could think of, for instance, the picture of a mosquito there, living organisms that some of them serve as vectors for disease. You may have heard, but the best hypothesis right now for COVID-19 origin is that it probably came from a species of bat. As species like that move around on the planet, then they can be vectors and bring diseases into different areas. And then the physical aspect of these agents could be something like, physical components of our environment, like the temperature. As the climate warms, like earthquakes, landslides, other harmful physical environmental factors. So we'll look at the chemical aspect of this in a little more detail. Just a few examples of some of the contaminants that we find out in nature. We mentioned PCBs back in the beginning of class with our Alaska salmon case study. It's a chemical that is used in manufacturing, right? Talked about it's used in insulators, in paper production, all these sorts of things. And environmental effects is a carcinogen. DDT is a chemical we're going to talk a little bit more today. It's a pesticide, something that we put on our crops to kill insects. And the first thing we found out about it was that it leads to thinning of eggshells, which can cause inability of chicks to fledge, right? Chicks to develop and hatch out properly. And then mercury is a contaminant which is found naturally in coal in a lot of places, and so as it's burned in these power plants that generate electricity, it goes up into the atmosphere, kind of like PCBs, comes down again with precipitation, and this is a neurotoxin, and so this is something that affects the brain negatively. You might have heard of mercury. Sometimes they'll have fish advisories. They'll say, oh, you can't eat any fish in this lake, especially large fish, because of high mercury contaminants. And it's most concerning with younger folks, children that are still developing their brain systems. Okay, so any of these chemical contaminants, there's reasons that they're dangerous, and there's really three factors that can. It's that they persist, they bioaccumulate, and they biomagnify. And we're going to talk about each of these separately, so don't feel that you have to jot them all down right now. But there's really these three different characteristics that could make these dangerous. So if we think first about persistence, right? So for instance, thinking back on PCBs in that first case study, we think about the fact that they persist in nature. And we might ask ourselves, why is that? Why aren't these toxic chemicals? Why aren't they just, transformed into something else, right? Why don't they go away? Why don't they react with other chemicals and go away? This is a graphic from lakes in Alaska, and it just shows all the different kinds of PCBs that are found in the sediment. So where it says PCB congeners on the x-axis. Each of those notches is like a different species of PCB, if you will. And so there's lots of different PCBs there, and they don't change. So we might ask ourselves, why do they persist? And the reason is because they're not a part of any of those biogeochemical cycles that we talked about earlier. So if you remember, we discussed nitrogen and phosphorus in their cycle, and the idea that they were taken up by plants as nutrients, right? And so they're transformed, they move through nature, they're used by organisms. But these contaminants are not part of any of those cycles because they're novel compounds, they're new. They were made by people, and before humans made them, they didn't exist, so there are no reactions in nature, in our ecosystems, that transform them. So they just stay out there, they just persist without transforming into something else. All right, so let's think about bioaccumulation for just a moment here. And this simply means being taken up by living organisms, getting into living tissue. And to think about why this is, you need to think about what your body does with chemicals that you ingest, but that it doesn't use, right? So for instance, if you consume more carbohydrates than your body needs, it stores that as fatty tissue, right, which can be broken down later on and the energy extracted from that. So the same thing happens with these toxins, that when they're taken up by organisms, it's stored in animals in the fatty tissue. And so because of that, these chemicals, for instance, if an animal is drinking water that has PCBs in it, those PCBs will get taken out of the water and transferred to the animal's fatty cells. So this is why they accumulate in living organisms. And then let's think about biomagnification. So biomagnification is the idea that as we move up higher and higher to higher trophic levels in the food web, the concentrations of these contaminants increase. So this is a schematic from the Atlantic Ocean ecosystem off the coast of Canada, looking at PCB concentrations. And so you can see in the phytoplankton, these small plants that float around in the water, PCB concentration is a little less than 3 parts per billion. Now zooplankton, interestingly enough, about the same, a little bit lower for some reason. But then we start to see the fish that feed on the plankton, so herring, they're feeding on the zooplankton and the phytoplankton. And now we look at the PCB concentrations in herring flesh, and it's an order of magnitude higher. It's 6 to 45 parts per billion. All right, well, seabirds feed on the herring, as do porpoises. And so in seabird eggs, we find PCB concentrations on the order of 3,000 parts per billion. Okay. Well, what about an organism that lives even longer? What if we look at porpoise tissue? And so in porpoises, we find concentrations on the order of 11 to 17,000 parts per billion. So you can see we find concentrations of toxins in these porpoises that don't exist in nature, but it's because they've eaten all these herring, which have eaten all this plankton, and as we all know, the energy moves up through that food web and it gets metabolized, it goes away, but the toxins don't go away. They keep getting stored in some concentrations get higher and higher. In fact, in some parts of the world, marine mammals, when they wash up dead on the beach, have to be disposed of as toxic waste because levels of contaminants are so high in the tissue. So it has to do with this pyramid of numbers, right? Where we have a lot of energy being produced at the bottom, right? And then it's moving on up. And as the energy moves on up, a lot of it goes away. It's metabolized. But whereas all of the energy at the bottom of that pyramid of numbers is not found at the top, all of the toxins in the organisms at the bottom of that pyramid of numbers are found at the top. And so that leads to higher and higher concentrations of toxins as we move up in the food web. Just an example of the importance of these toxins and how they can affect species. So bald eagles were one of the first species listed under the Endangered Species Act in 1973. And so the blue line there is the average number of young per breeding area. And the red line there is DDE residue, which is essentially DDT residue in eagle eggs. And so in 1973, actually late 1972, DDT was banned, right? You could no longer produce it. So sure enough, the amount of residue in eagle eggs goes down, the mean number of young per breeding area goes up. DDT was a problem led to problems in egg development of birds. Now something else happened in 1973, which you might be able to tell me. That was the Endangered Species Act. I guess I told you that already, actually. And so, there are other things that were done to conserve bald eagles besides banning DDT, which no doubt contributed. But still, DDT has been shown to have harmful effects on bird eggs. So I just wanted to share a story about DDT. And I think it's interesting because it really illustrates the way that our opinion about a new innovation can change over time. And in some cases, we can make a 180 in our opinion about it. It also is kind of a case study in how science works and how that knowledge gets transferred. So DDT, again, it's a novel compound. It didn't exist in nature until it was made in a lab. So 1873 was when it was first synthesized. And, organic chemists like to make things, but there were really no notable qualities. They were able to make this new molecule. It's interesting, but nothing really significant about it. So fast forward to 1935, and so scientist Paul Muller was screening different chemicals for insecticidal properties. In particular, he was looking for a chemical that could combat Colorado potato beetles. These were a real severe agricultural pest. Well, it turned out that DDT was very good at killing these potato beetles, so a sample was sent to the U.S. in 1942, saying essentially, hey, here's this chemical that looks like it's great at killing these insect pests. So the 1940s, of course, were the time that World War II was going on, and the U.S. military was also researching potential pesticides. In World War I, 2.5 million soldiers had died of typhus. And so when soldiers were marching through a new area, they were sleeping in places that weren't safe. Typhus is transmitted by lice. So it was really a military priority to find chemicals that could be used to kill these insect pests to help with survival of soldiers. So in 1943, research was done, and DDT was shown to be 100 times more lethal to, in this case, mosquitoes than any other agent. So DDT was very good at killing these insect pests. So then there was a typhoid outbreak in Naples in 1943. DDT treatments were used and it was very effective at killing the lice. Now, what DDT does is it messes with the chitin, which is a structural carbohydrate molecule that insects use to construct their exoskeletons. So when you use this chemical, insects are not able to build their exoskeletons properly, and they die. So World War II was the first war in history where more soldiers died from battlefield wounds than from disease. So lots of other technological innovations came out of World War II, but DDT was one of them. So in 1948, Paul Muller receives the Nobel Prize in Physiology or Medicine for his discovery of the insecticidal properties of DDT. And so in the citation, it's noted that DDT is cheap, easily manufactured, and exceedingly stable. Surface treated with DDT maintains its insecticidal properties for a long time, up to several months. Right? So that was a great thing, right? Was how stable this chemical was. It goes on to say the story of DDT illustrates the often wondrous ways of science when a major discovery has been made. Right? This idea that he was working with flies and Colorado beetles discovers the substance that is effective in the battle against the most serious diseases in the world. So DDT at this point was kind of like the wonder drug. It was seen as a safe new pesticide, revolutionary, could reduce disease by killing these insect vectors, and could really help with agricultural production. Remember, we're starting to get into the Green Revolution now, right? This time period when our agricultural production across the world started to skyrocket, and the use of these chemical insecticides was a big part of that. So the picture on the left actually shows a beach in New Jersey where they would drive down the beach and they would fog the beaches with DDT to kill mosquitoes, because you don't want any pesky mosquitoes messing with your outdoor fun. And thought to be completely safe. So these women in this picture are completely fine to have this DDT sprayed all over them. And so it was even recommended for use inside kitchen cabinets to fight against roaches and other bugs, that kind of thing. So this is a U.S. Department of Agriculture circular as a picture here where it says DDT is good for me. Everyone was singing the praises of DDT. Now, in the late 1950s, there were some negative effects of DDT that started to be noted. Rachel Carson, who we talked about a little bit earlier this semester, was a science writer for the government. She wrote articles in magazines and newspapers talking about scientific issues. So she was fairly well known and had a following. And so people started writing into her with these letters about seeing primarily birds that died in areas after they've been sprayed with DDT. So the evidence started to accumulate about the potential negative effects of DDT. So in the 60s, the book that Rachel Carson is best known for, Silent Spring, is published. And President Kennedy, who was president at the time, read an excerpt of the book, which was originally published as a series of articles in New Yorker magazine. And he assembled a commission to study this problem, to try to figure out how much of an issue is this. Well, there was opposition. The companies that produced DDT, argued that DDT was required, that the use of these insecticides was required for us to produce agriculturally at high levels. Someone from the American Cyanamide Company, was a pesticide company, writes, "If man were to faithfully follow the teachings of Miss Carson, we would return to the Dark Ages, and the insects and diseases and vermin would once again inherit the earth." So definitely some fear-mongering here, trying to get people thinking that we were not going to be able to survive without these pesticides. But lo and behold, come 1972, as I mentioned already, DDT was banned in the U.S. Now, it wasn't banned globally, and it is still produced in some parts of the world. And interestingly enough, the World Health Organization still advocates using DDT to kill mosquitoes in areas where malaria is endemic, right? So we'll talk in just a second about the negative health effects of DDT. But clearly, malaria kills over 1 million people a year globally, and it's a health threat which also has to be managed. To show you a little bit of data on there, this is an article that looked at DDT use around the globe and how it related to malaria. And so Mexico uses quite a bit of DDT, or used quite a bit of DDT, over twice as much as the closest country in South and Central America, which was Brazil. And so if you look at this figure, it has the number of households sprayed with DDT there in the blue bars, and the number of cases of DDT in the black line. To me, it's a story here, moving from left to right. Initially, lots of households sprayed with DDT, very few cases. as the number of households being sprayed declines, start to see an increase in the cases of malaria. Then in the early 70s, when we see this peak in DDT cases, there are more households that are sprayed, and as those number of households sprayed increases, again the number of cases drop. Then as the number of households sprayed gets down really low again there in the 80s, there's a spike in the number of cases again. So, DDT is very good at killing mosquitoes, and that helps with malaria prevention. And I'm not trying to say that DDT is the only way you can fight malaria, but it is effective. And like I said, the World Health Organization still advocates using it in certain areas where malaria is a big problem. But they advocate spraying it a little differently than we used to. We used to spray it all over the fields. Now they just advocate spraying it on the inside of homes, where it won't be in the air, where it'll coat the walls, and then that'll kill mosquitoes that would stay in those homes during the day and then come out at night and bite people. All right. Well, here are the human health effects that we know of as far as DDT goes. It also is a carcinogen, has been linked to increased prevalence of certain cancers. It's an endocrine disruptor, so our endocrine system relies on levels of hormones where very minute changes in the concentration of hormones have systemic effects over our whole body. DDT, like a lot of agricultural chemicals, has a structure that is similar chemically to estrogen, and so it mimics the hormone estrogen in people's bodies. And then when there's chronic exposure, you see things like damage to our nervous system, psychological effects, asthma, and then it's also been linked to type 2 diabetes. So none of this was known, of course, back when Paul Muller earned the Nobel Prize. So these animals are changing their tune. Cows aren't quite sure that they should be singing the praises of DDT anymore. All right. So we have these toxins out in the environment, and we can be exposed to them in different ways. We can suffer acute exposure or chronic exposure. And so exposure is when we have high levels of exposure for short periods of time. Chronic is when we experience low levels of exposure for longer periods of time. And this is about the time when I'd ask you which of these is easier to document and which of these is easier to study, right? And I think you'd all probably say exposure. The problem with chronic exposure is that the effects don't show up until much later on in life. And, so if there's a higher incidence of cancer with people that have been exposed to DDT over 20 years, the problem is there's a lot of other things going on in the life of those 20 years as well, and so it can be real hard to tie a causal, a causal mechanism to DDT. So again, with chronic exposure, cause and effect may not be easily apparent. It can be hard to figure out. These more gradual effects throughout our whole body systems, affecting organs like the lung, the liver. But often when we learn about the negative impacts of chemicals, it's from acute exposure events, right? So we know that crude oil is bad for marine organisms because we've seen the effects of massive oil spills in different parts of the ocean. And so we look at that and we can say, okay, it's a pretty good bet that exposing marine organisms to low levels for a long time is also going to be bad because they affect them negatively from an acute exposure perspective too. So chronic exposure is much more common but harder to detect. Acute exposure more rare, but cause and effect is more easily evident. So I mentioned the endocrine disruptors, right? So hormones, again, these chemical messengers that our body makes in really, really tiny amounts. And, they regulate the whole way that our bodies work. Obviously, development, sexual maturity for men and women is the clearest example, right? where whole body systems develop differently once you have changing concentrations of certain hormones. But what's interesting is that there are several pollutants, including DDT, PCBs, and bisphenol A, BPA, which you might have heard of, that are, again, function as estrogen mimics. And so because of that similarity in structure, our bodies often treat them as if our body has been releasing that hormone. So with these endocrine disruptors, for instance, there is some debate that declining sperm count in men is a consequence of this. Some data on the upper part of the panel there from the 40s into the 90s showing a significant decline. Similarly, increasing incidence of testicular cancer has also been hypothesized to be due to increased levels of endocrine disruptors, again, in particular chemicals which mimic estrogen. So, if you think as well about all of the drugs that people take, right? All the prescription drugs, our body doesn't process all of those, and so they can leave the body in the urine and get into our wastewater treatment systems. In addition, we're told to dispose of prescription drugs that we don't use, and one way that people do that, although you're not supposed to, is by flushing them down the toilet. So we have all these different drugs that are in our water bodies, and this is an interesting study that found traces of antidepressant drugs in bluegills in Texas. This is my mandatory fish story of the day. So anyway, so the chemical is right. So this came from the city of Denton in North Texas, and comes into this creek. So you might wonder, what does it mean that Prozac is being found in bluegills? The author says, fluoxetine and other antidepressants affect fish in roughly the same way they affect people. It relaxes them. And so we've got some fish that are a little more relaxed than they used to be. But then the author says, maybe it makes you a happy fish and you're kind of hanging out. But how does that influence your ability to capture prey? Do you instantly become candy for largemouth bass, which is a predator, because you're accumulating large amounts of Prozac in your system? These are areas where more research is needed. So maybe we have some real mellow fish out there that are easier for bass, for their predators to eat. And then maybe their predators become really mellow from getting antidepressants from their prey fish. So these are all things that we need to learn more about, and we just don't have that much data on yet. Although, again, this particular study is about 18 years old. All right, so if we're trying to manage these chemical contaminants in the environment, there's a couple of steps that we need to go through when thinking about any kind of risk. The first is what's called risk assessment. So you need to figure out how risky a particular action is. For instance, consuming fish that has been contaminated with Prozac, for instance. And so you need to use statistical methods to do this, and you basically calculate the probability of a given risk. So just to throw some data at you, if you look at different causes of death for us and our 1-year odds and lifetime odds, cardiovascular disease, right? The number one killer in North America, lifetime odds of 1 in 4 dying from that. Motor vehicle accidents, 1 in 88. Commercial aircraft, 1 in 40,000. So the first thing you need to think about whether eliminating a risk is just how risky is it. So, motor vehicle accidents quite risky, right? If 1 out of 88 people is going to pass away from motor vehicle accidents, it's certainly risky, but then you have to balance that with the benefit. And certainly we have lots of benefits of being able to get around in motor vehicles, although it's nice to walk and bike as well. And so then we think about how to reduce those risks, right? So cardiovascular disease we can reduce by exercising, eating right, all those sorts of things. So then the question about whether that risk should be eliminated is the risk management piece. So risk assessment, how risky is that activity? Risk management then, does something need to be done to eliminate that particular risk or not? And this is where the potential benefits of that come in. And there's two extremes to a spectrum here when it comes to managing risks. You can have the innocent until proven guilty approach, or more of what we'd call a precautionary principle approach. So as the name implies, the innocent until proven guilty approach essentially says, until we find a problem with this particular activity, we're going to assume it's just fine. Whereas the precautionary principle approach takes the approach that we're going to look for that problem really hard early on. And so your book has an example here of two chemicals being brought to market. And so there's industrial research and development, pre-market testing, and then it gets to the market, consumer use of the products. Post-market testing, and then regulation and bans of unsafe products, right? Or like a recall, like you might think about with like a car, for instance. And then the final step is consumer use of safe products. So a precautionary principle approach is going to lead to a lot more rigorous testing before they reach the market. Whereas the innocent until proven guilty approach, post-market testing is going to play a much bigger role. All right, there's going to be testing going on after a problem is identified. I think it's interesting to think of this this dichotomy relative to GMOs, because for a long time Europe had a very strict policy on GMOs. It's lessened a little bit. And it was based on very much a precautionary principle approach. They said, look, we haven't found any negative effects of GMOs yet, but we need more testing to search for them. Whereas in the USA, again, as we talked back before break, I guess, man, that seems like a long time ago. In the U.S., we had no regulations on GMOs, and will not have any until next January. So the U.S. took this approach of, hey, we haven't found any problems with GMOs yet, bring them on the market. So the interesting philosophy in those two different places with regard to GMOs that it illustrates Europe taking more of a precautionary principle approach, and in the U.S. more of an innocent until proven guilty approach. All right. Well, I hope you guys are doing well. Again, let me know if you have any questions at all about lecture or other materials as well. Be